So I think Ross introduced you to phylogenetics, so you should all be experts, right? You should all know all about phylogenetic trees and that kind of thing. But basically, a phylogeny is a diagram that shows evolutionary descent of, it can be the same species or different species from a common ancestor, so how much they've diverged from a common ancestor. So if you have a tree, I'm sure everybody here in some context has seen a phylogenetic tree, whether it's a family tree or any other kind of tree. And I'm sure that you're familiar with this, but I'll just go over it really quickly. So you have, in this case, we're mainly talking about bacteria and viruses, but these would be different individuals. So it could be people, it could be bacteria, it could be viruses. So here we have 4 individuals, and they're at the tips of the tree. These are nodes, where the branches connect are nodes, and they would represent a common ancestor that we don't have information on. So it would have been a common ancestor in the past. And these are branches of the tree. And the branches are the important part of the tree. They show the distance that these different individuals have from each other. And in this case, this tree has a root. Not all trees have a root. So one important thing about phylogenetic trees is that the order of the tree doesn't matter. So the order that these are in doesn't matter at all. So if you look at these trees down the side, can you see that they're actually the same tree? Yeah? Do you see what I mean? Even though they're in a different order. Because it's to do with the nodes, what connects the nodes, and the branches, so the order of the branching rather than the order of the tips. Because you're just flipping things around to look at them in a different way. But as I say, these are absolutely identical trees. And that's quite an important thing for when you interpret phylogenetic trees. Okay, so in order to build a phylogenetic tree, certainly, you need some kind of sequence data. In this case, it would be a genetic sequence data. You need a model of nucleotide substitution, which I find a bit complicated, but I'll go over it really quickly. And you need an ability to determine this branching order, so to determine what the nodes are and how these are related to each other. So often phylogenetic trees are made with nucleotide data. You can also do it with amino acids, that kind of thing. And the way that we do it these days often is from sequencing a pathogen. So you can do whole genome sequencing and look at the relatedness of the genomes. This is really easy for viral genomes because they're quite small, but it can be quite complex for bacterial genomes because they tend to be a lot bigger than viral genomes, and they have a lot of differences in terms of gene presence and absence. So I don't know how much you know about bacterial evolution, but they have horizontal gene transfer as well, where they can actually exchange DNA with each other. So in terms of a phylogenetic tree, that would affect the tree because they're exchanging DNA with each other rather than just evolving the way that we evolve. You see what I mean? But once you get some sequence that you're interested in, whether it's a gene or a genome, you need to actually look at the distance between the sequences. So you know when I was talking about the multi-locus sequence typing, and you have these small regions and genes that you're looking at differences, these are bigger bits of sequence. But again, you're just looking at the distance. So you just look and you say, at this point, these are all A's, but this is a G, so this is a little bit different, and you work along like that. And so you're looking at variable sites. So if you look here, this is a piece of software that you're going to look at in a minute called Clustal, and it's got a multiple sequence alignment, but it's not actually aligned very well at the moment. So you can see that these particular genomes here are quite well aligned. You can see that all the letters, the A's, T's, C's and G's, your nucleotides seem to line up quite well, but these seem to be quite messy. But if you look, you can see that this part here probably is the same as that. It looks the same, it's just not being lined up. So you need to do some kind of sequence alignment to do this. And I mean, yeah, you could do it by eye, but it would take quite a long time to do, and I wouldn't recommend it. But when you actually use something like Clustal to do a multiple sequence alignment, you can see that this, which is quite messy, has gone to this. And so the parts that you're interested in, the parts that define the branches in the tree, are these variants here. So you can see some parts are really conserved, like this A down here is the same all the way down. So that's actually not very interesting because it's the same in all of your sequences. But some of them seem to have a G here instead of an A. And so that's what makes them variable. That's their variance. And then there's also gaps as well.